Every single cell in your body has a copy of your entire DNA, every one of which can stretch out to approximately as tall as Darth Vader. Every cell that dies, that carries with it the code to your entire DNA, and with it, the need for another cell with the same complete set of DNA. This requires DNA replication. From your heart to your skin to your liver, DNA replication is the only thing keeping you and me alive. But how does it work? If you look it up online, you'll get either exceedingly simple explanations that tell you almost nothing about it, or extremely boring videos filled to the brim with words that seem to have been made with the distinctive goal of confusing you. That's why I've made this podcast, to explain it in a way that you don't actually need a degree or to spend the next month of your life researching how DNA replication works. That's what I've done. Before getting into the how, we have to understand two things. First, that DNA is made up of different nitrogen bases called G, C, A, and T. Now, they have actual names, but literally no one ever uses them. Each of these nitrogen bases are each part of a 3-molecule set called a monomer of DNA, more commonly known as a nucleotide, although I don't know how commonly either one of those terms is. The nitrogen bases, which I will now be calling letters from now on because it's easier, can only fit with certain other letters. T can only fit with A can only go to T, G can only go to C, and C can only go with G. This is due to the chemical size of the letter molecule or something. Just trust me as I trust the scientists to figure this out. It just works this way. If you want to do your own research, have fun with the next month of your life. Now, the letter pairings of T and A, etc., are stacked on top of each other via the other 2 molecules in the nucleotides. This makes the end product look like a ladder, at least when it's all stretched out, that is. For most of its life, it will be twisted onto itself until you get the double helix shape we are all used to seeing in regards to DNA. We are now ready to get into the details of DNA replication. You have enough backend information here as I care to give you, so we can start actually learning what I want to teach you. DNA replication starts with something in the cell that tells it to replicate. I'm not going into that. I just want to talk about the physical replication of the DNA itself. After it receives its order to replicate, it starts off by having to untwist and pull the 2 strands apart from the DNA helix. It does this by having the topoisomerase, from now on in this podcast called the DNA topo, for the ease of speaking, cut and untwist the DNA helix into the DNA ladder. In time with the DNA topo, its partner, the DNA helicase, just sort of pushes through the letters, breaking them apart. We are now left with 2 mirroring strands of DNA. Now, while the DNA topo and helicase are going, the nucleus enacts a DNA lockdown, stopping all action in the cell that is not a part of the DNA replication. This lockdown does not, however, apply to DNA nucleotides. Both the DNA and RNA nucleotides are from the plants and animals we eat every day and are practically free-floating in your cells. The DNA polymerase is like a DNA zipper. It holds on to an already completed part of one of the new ladder strands, then guides the chemically magnetized letters together and binds them together to continue the new DNA strand. However, like a zipper, it needs something to get it started. It can't start off with nothing on one side. So there are about 9 to 10 nucleotides of little gibberish at the start of the DNA strand. That gibberish is RNA, which is basically DNA that has a different ladder base. The sides of the ladder are different, and it has a different letter. Now, in order to put the RNA onto the gibberish for the polymerase, we use an enzyme called the primase. The primase acts as a sort of mold for the RNA primer in gibberish. The primase allows the RNA to fall into place so it can bond the primer and the gibberish together. All of this allows the polymerase to actually start creating, or shall we say, replicating, a new DNA strand. The polymerase 3, because yes, there are at least 3 of them, goes along in a zipper-like fashion, reading the original strand and picking out the correct nucleotides based on the letter, binding it to the original strand. The nuclease goes in and looks for any obvious quote-unquote kinks and mismatches. The nuclease tells the polymerase II when it does find something wrong, and the polymerase II replaces the incorrect nucleotide. This is the full extent of semi-conservative DNA replication. However, in DNA, there is always a way up and a way down. In order to connect the two strands together, we need to flip one of them upside down. This is obviously a metaphor, but it's easier than the facts while staying the truth. After all, you're not getting tested on this. The side of the strand that is facing down and the new strand therefore is facing up is called the leading strand, and that's the one we've been talking about. The other strand that is facing up and therefore needs the new one to be facing down is called the lagging strand. The leading strand's polymerase III is moving from the gibberish towards the helicase, moving and adding to the DNA strand in the same direction the DNA strand is coming from. The lagging strand, on the other hand, replicates going away from the helicase. This means that one polymerase can't replicate the entire strand like in the leading strand. Instead, we have to replicate in small fragments of 150 to 200 nucleotides, called Okazaki fragments. These fragments are made by having a primase create an RNA primer, followed by a polymerase III replicating the DNA from the RNA primer to the gibberish or an earlier primer. Now, don't let me confuse you. I'm using gibberish and RNA primer interchangeably to talk about different orders of the strand. They're both RNA primer. They're both gibberish that we don't use in order to tell ourselves what to do. After the polymerase III reaches the gibberish, it breaks off the Okazaki fragment without being able to connect the latter sides of the DNA with the latter sides of the RNA primer. The polymerase I then comes into the gibberish and replaces the RNA primer with DNA nucleotide. The ligase then connects the latter sides of the DNA that was RNA primer with the end of the newest Okazaki fragment. Then the whole cycle repeats repeatedly for the lagging strand until it finishes replicating the whole strand. Then, like the leading strand, the nuclease comes in, rips out any inconsistencies, and has the polymerase II fix them. But what happens to the DNA topo and helicase when they reach the end of the strand? Do they let go of the strands and let them fly apart uncontrolled, or do they hold on, blocking the polymerase III's from finishing the strand? Of course, they don't do either of those options. Instead, there are about 1,500 to 2,000 nucleotides of complete gibberish, true gibberish, if you will, called telomeres. This allows the DNA topo and helicase to maintain the control of the DNA strands while still allowing the polymerase III's to replicate the DNA. After the polymerase III does its job and replicates all the DNA, we are left with the telomeres hanging off the DNA. In order to have something for the next DNA topo to hang on to, the telomere loops in on itself. This shortens the telomere, shortening the quote-unquote DNA timer, that is the DNA telomere. Some cells are able to repair the telomeres. These cells have a telomerase. The telomerase adds more DNA nucleotides onto the telomeres in a repeating pattern of letters that only the telomeres have. And that was semi-conservative DNA replication in a nutshell in about 8 minutes.